we're very excited to be part of it, and I'm very relevant to what has been talked about throughout this entire meeting. So I'm talking about a project with short-term benefits of caloric restriction. So as mentioned earlier in this meeting several times now, that CR is one of the most well-established forms of an aging intervention. It's been known to extend lifespan and delay aging for an extended period of time now, for almost a century. And now all of these variations of this caloric restriction have come about in multiple model organisms, whether we're looking at intermittent fasting, these fast-mimicking diets, ketogenic diets. It's almost embarrassing to be saying this, as the people that have done these and started all of this are here in the audience. But essentially, we're finding various ways of doing this intervention, and we're finding positive benefits in many of them. However, the question is, what is the translatability of this? How do we go from a CR that looks fantastic in model organisms into humans? And so there are beginning to be these studies where we see this. Dr. Dixit gave a really nice overview of the calorie experiment, where they do, I believe, 25% restriction for 2-year periods. But I think one of the biggest challenges that we have is that we as humans, as much as we like to think that we have pretty strong motivation, it's really hard to adhere to these restrictive diets. So can we find ways that are maybe a little bit more feasible than constant restriction? We like the time-restricted diets because it allows us to eat sort of whatever we want for short periods of time. Do we have sort of this light at the end of the tunnel instead of every day waking up and being like, oh gosh, I really wish I could have that extra 20% calories, but you're restricted? So can we find a way to also have mental positivity in what we're doing, not just with the hopes of these positive benefits mechanistically? So of course, as Dr. Anderson beautifully talked about with the caloric restriction, there have been several studies in non-human primates, which is our best intermediate model to look at these interventions and how they may affect humans. So there have been several studies that really all are positive, suggesting that caloric restriction really does improve health span and lifespan in these non-human primates. However, even within these 3 studies that are really landmark studies, there are variations between these experiments. And coming from a mouse model where you're already concerned about variation in the mouse model, moving into this heterogeneous population of non-human primates, in our heads really sounds exciting, but in practicality it becomes quite difficult. So all of these, like I mentioned, do have these positive benefits with slight differences because there are slight variations. But essentially what we're taking advantage of is this beautiful work that was published by Alberto in Rafa de Cabo's group, where they did this 4-10 study. So it's not lifelong caloric intervention. It was an intervention that started in midlife, so it's not something that you needed to have started when you were young and not thinking about how you want to maintain your longevity forever. So middle age, when most of us start thinking about this. And it was a 3-day or a 4-day diet. So the first day, the diet was 50% of what they would normally eat, and then 3 days of relatively restricted diet, where they were only eating 30% of what they wanted. But then for the next 10 days, they could have at it, they could eat whatever they wanted, and they did this in repeat cycles. And the way they had done this cycle was they did it for 5 months, so it was essentially 10 cycles of this period of feeding and then a period of eating. And what they saw is that they had a really nice decrease in body weight change. I'm sorry, I can't get my mouse to work. Okay, here we go. A decrease in body weight change, and it was sustained in these male mice. This was also associated with other positive benefits in metabolic fitness as well as physical fitness. And so we thought this would be a lovely model to translate into the non-human primates. We were very fortunate to get involved with this project that was really headed by Dr. Julie Mattson and Kelly Vaughan at the NIA's Non-Human Primate Core. And they put together this sort of extension of the mouse model I just talked about. So we had 23 total animals. We tried to balance them by age, at middle age, so about 14 years old. And we also included females as well as males in this study. And we tried to match them essentially by body weight and split them in equal groups. And essentially do the exact same diet as I just explained. So we measured their food consumption for 2 weeks, then a 50% reduction, 30% reduction, and then ad libitum feeding. And this seems relatively straightforward, right? 23 animals, a 2.5 month time period. But this really is a quite extensive experimental setup that I really want to highlight, because the work that Julie and Kelly put into thinking of these experiments, when we're like, oh, can you just do this? It's pretty amazing. So these 23 animals ultimately were split into 4 different groups. And these 4 different groups were followed for 2.5 months, and you can see the staggering of these. But these monkeys also needed to be individually maintained and watched, because unlike the mice, when you feed them small amounts and they don't really know what's going on, apparently if monkeys sense that other monkeys are getting food, then there's chaos. So they had to separate them early on and kind of prepare for months before this study actually happened. So it's really exciting to be involved in an institute or in these types of experiments where we can translate some of our data like this. Okay, so baseline experiments, and I'll try to highlight the people who've done the analysis going through. So this is Dr. Hagai Yanai, who's done most of this analysis. So essentially we start with food consumption, and between the 2 groups for 2 weeks, we trace how much food that they're consuming. Then we have the 6 cycles of dietary intervention, where you can see at this first time point, there's 50% food and it drops to 30, and then they return back to sort of their normal eating. So one thing that we were very conscious about was in the rodent studies, what happens is after they return back to this ad libitum feeding, sometimes they will gorge themselves because they've been restricted for 2 days, and they're like, oh, food, I need to eat everything. So there's a little bit of a spike, and then it returns down. And what we do see in these non-human primates, when we sort of averaged across this entire time course, is that we don't see that same spike. And even when they return to the ad libitum feeding, they still sort of maintain a slightly lower amount of food that's consumed. And this is similar to if you look at the very baseline measurements, that we had a slightly lower amount of food in the PRF group when they started with. So they returned essentially back to what they were eating at baseline, not overeating. And so throughout this time course, what we looked at were DEXA composition, so the body composition. And as Dr. Anderson mentioned, loss of fat isn't everything, but it is something that we wanted to look at and see how these body composition changes. So we looked at at the middle of the diet and also at the end of the diet. So cycle 3, day 14 is at the end of the cycle, so they've already been allowed to refeed. And cycle 6, day 14 is again at the very end of the experiment. So you can see at this middle time point that they're already beginning to have loss of both lean body mass as well as loss of fat. And again, all of these are in relation to their baseline starting points. And so what we were impressed with is that we had kind of this global loss of body fat. And surprisingly, it wasn't restricted to any particular area. It wasn't in more Android versus gynoid. I remember this by Android versus Mac, but thank you, Roz, I appreciate that. I tried to throw something in there and make sure you guys are awake, but so there weren't changes in that, or the trunk fat versus limb fat. So it was sort of this global loss of overall fat, which we took to be as a positive thing. So when we looked at the overall body loss, of course, there was this loss associated with the diet. But what we were particularly interested in is that it didn't directly correlate with the overall restriction in the diet. So if you can see here, what we did was the effective CR. So we looked throughout the study, averaging across how much they ate during the restricted period, during the time that they were ad libitum fed. And the ones that actually had only about 15% restriction were the ones that actually lost the most body weight. And those that were on the higher amount of restriction, so closer to the 30% restriction, were the ones that actually had the least amount of body weight change. So this was a little bit surprising to us, because you often think calories in, less of them, you'll have less body weight. But this isn't really what we were seeing in this experiment. Also, surprisingly, when we finished this experiment, these monkeys then returned back into the colony, and many of them are just kind of happily living out their lives in this non-human primate colony, and some of them were enlisted in other studies, none of them on dietary restriction. But we have almost 2 years of follow-up with body weight in all of these animals. And what was really striking about this is now these animals are back to ad libitum feeding, doing whatever they want, and the majority of these PRF animals have now maintained this lower body weight throughout this 2-year follow-up, slightly longer than 2-year follow-up. Seems fantastic, right? It seems like there's some sort of memory of this diet. Well, here's the bad news, or maybe good news, depending on if you're male or female. So what happens is in the females, after this diet restriction, they managed to almost immediately regain the weight that was lost and come back to a baseline level that was similar to their ad libitum fed counterparts within the colony. The males, on the other hand, once they lost this weight, it was almost like they had completely reset. And now, even though they're doing whatever they want, they never sort of get that body weight gain that's associated with the aging of the animals and the rest of the colony. So we really begin to see this sex divergence with this pretty short period of intervention between the males and females. So we wanted to perhaps understand what might be underlying some of these changes, what could be driving, what could have these long-lasting effects. And so we were fortunate to enlist Dr. Aoun to help us with looking at some of the serum changes of metabolomic changes in the serum. So we tried to find some relatively non-invasive procedures to collect information on these monkeys, since we're keeping them in the colony longitudinally. So these are blood serum collections that we did peak diet cycle in the middle, and then at the end, peak diet, and also after they've been refed. So just looking at some of the differential abundant metabolites in the ad lib on the top and the PRF monkeys at the bottom, I hope that it's relatively abundant or relatively clear in these volcano plots that there's really a lot of changes occurring in the PRF monkeys and some changes that are occurring in the ad lib monkeys, but this is sort of just things that are occurring during natural time. And so we've accounted for these as well, but surprisingly, 10 days later, after the 6 cycles, if you look at the metabolite changes in the bottom at the PRF, you can see that there's really not a lot of changes. So it looks like these metabolite changes are short-term, they occur robustly, and then they may go away. So when we do principal component analysis of these changes, that is indeed what we see. We see these consensus shifts that are occurring at the peak diet. So you can see the red and dark red dots have sort of shifted away from the middle. But then once after the ad libitum feeding, they all return back to sort of this baseline, or what the ad libitum metabolite profile looks like. So we wanted to dig a little bit deeper into what was being shifted during this dietary restriction time. And so we split this now into the males and females, cycle 3, day 4, and cycle 6, day 4. And I hope what you can appreciate here is that we have a lot of red here in the lipid class. So it really looks like we've shifted from a carbohydrate metabolism to a lipid metabolism during this restricted period. And when we essentially do the overlap between things that are upmodulated in cycle 3 and cycle 6, so what we believe are kind of the consensus marks of the caloric restriction, really the upmodulated metabolites have to do with fatty acid synthesis, essentially lipid metabolism. And this was nicely correlated with what we saw in the murine model as well. Unfortunately, when we now look at what happens when they've been refed, and this is in comparison to baseline, the males essentially shifted back to what their baseline levels were. They weren't lower, they were pretty similar to what happened originally. But if you look at the females, especially in this lipid section that had become upregulated, they essentially down-modulated this. So what we believe is happening is that they've overcompensated almost for this restricted period and are now holding on to their lipids as much as possible here. And we believe this is sort of what's contributing to that response and the body weight gain that occurred quite rapidly after they went back on their normal chow, and that the males had sort of maintained this lower amount. So we'd sort of essentially reset their metabolic profile, and that's lasted over an extended period of time. So we also know that caloric restriction has been known to affect the microbiome. And again, as this is another accessible piece of information that we can collect, and I like to say it's accessible because I was not the one collecting the feces, so thank you to everybody in the primate colony. But we collected these, again, throughout the time course, and we wanted to see how this diet might affect the microbiome. So I honestly thought that there wouldn't be changes. The monkeys are eating the same things, they're just eating less of it, but the diet composition is exactly the same. So this was work done by, the analysis was done by Bong-Soo Park and Bob Koo, who's so Bong-Soo is in my group at the NIA, and Bob is at SUNY in South Korea. So the first thing that we looked at were sort of basic microbiome analysis, where we looked at alpha diversity. And alpha diversity is essentially the measure of diversity within the group. So how much diversity do these monkeys who have undergone the diet restriction have compared to the ones that are just eating normally? And usually we associate with increased alpha diversity with good things, decreased alpha diversity being not as good. So hopefully what you can appreciate here is that at cycle 6, day 4, so end of the cycle, middle of the diet, we have increased in alpha diversity in the restricted animals. And this was when we compared the restricted to the ad libitum, but we also see the same benefits in the increased in alpha diversity when we compare the animals to their baseline. So it's not just the group as a whole, but the group compared to its baseline. So we really do think that these benefits are occurring. And here at the cycle 6, day 8, which is when they started refeeding, they still maintain sort of this improved alpha diversity. So we're really thinking that this may be a potential mechanism in which this resetting is occurring by affecting the microbiome. When we looked at beta diversity, which is now the comparison between the groups, so not just within the individual monkeys, but between the groups as a whole, at baseline we had no real differences in beta diversity, again saying that our populations started out relatively similar. But once we looked within the peak cycles, we can start to see a small separation between the beta diversity, where you really see the groups in red separating up away from the ad-libs. But something that I hope is appreciable to all of you as well is that there's a lot of heterogeneity in these data. So the number of monkeys required to see kind of these small differences is quite large. But we were happy to see that we have this change. And then we wanted to see what kind of alterations might be happening. What are the taxa that are changing? Are any of these suggestive of positive benefits? And indeed, what we see in these differential abundance analyses is that there's an increase in the Verrochia microbia. Please forgive me, I didn't take Latin, or in the lentisphera. And so for some of you out here, especially Cott, he loves this Akkermansia, is that one of the microbiome bacteria that's positively associated with aging, and healthy aging is one of the main contributors to Verrochia microbia, which is the Akkermansia. You can also see that the firmicutes goes down, and we also associated this with positive benefit because firmicutes also go up in diabetes and obesity. So we were seeing sort of these glimmers of hope that the changes in the microbiome may be indicative of positive things occurring. And we can also see that these are transient but cumulative changes. So again, looking at PCOA plots, again, you shift from the center with the black and the dark colors, and then up to the right, and then down to the left. But the cycles are associated with each other, the days are associated with each other, not the cycles. So it's suggestive that during the diet, we have kind of similar changes that are occurring in the microbiome. And then you can see on the panel here, if you look at, I'm so sorry, I should be using my pointer here. If you compare here and here, cycle 3, day 4, and cycle 6, and really looking at this median line, you can see it looks like there's a more robust response that happens on the 6th cycle versus the 3rd. So it happens in the same pattern, but it seems to be a little bit of a memory and a cumulative build. So the response occurs more robustly the more times you do it. So we have positive benefits in the microbiome, changes in metabolites, and here's actually how we became involved in this study. We were interested in the changes in the blood. So as Dr. Dixit had mentioned, one of the things that is most commonly associated with these positive changes with caloric restriction is this reduction of inflammation. So we thought we could maybe see changes in the composition of the blood that may be associated with some of these positive benefits we are seeing. But unfortunately, what we saw were not really big differences that happened. So we actually had reached out to Dr. Anderson, and we were talking about this data. And we're like, there's not a lot of change, but it looks like then the PRF monkeys change even less. She's like, well, that's interesting. And we're like, oh, okay, great. So what we can see here is that actually the blood seems even more stable in these PRF monkeys. We don't see positive changes that we were hoping, sort of improvement in the lymphoid cell types or decrease in the cytokines. But you really do see that it's more stable. So this is suggestive that this could be a positive benefit. It's just not associated with changes in the composition, nor with changes in the cytokine levels. We did, however, see some very small differences in unsupervised analysis of our flow cytometry data. So potentially populations of cells that we don't typically look at appear to have changes. Some of them are unknown T, or a subset of T cells that we're not sure of, as well as a subset of CD16 cells. Okay, so blood changes stable, microbiome positive, metabolome good. I'm just trying to bring you along with me. So ultimately, we wanted to see, do these changes in the metabolome also have changes in, or the changes in the microbiome, are they similar to what we see in the changes in the metabolome? And so it's shown here is essentially at day 4, so peak diet of all of these, what are the changes of the microbiome, and do they happen to have any sort of implications that they may be contributing to changes in the metabolome? And again, the highlight here is really just to say that these things that are enriched on day 4, multiple times throughout the diet, really seem to be involved in lipid-related pathways. So again, even though loss of fat is not necessarily completely a positive thing, we are associating it with this positive benefit that we're seeing in these non-human primates. So we have this concordance between the metabolome and the microbiome changes. And again, we see a very sex-specific difference in the enrichment here, in which the male monkeys have changes in the microbiome that are unique. The females under the PRF diet only have changes that are also found in the ad libitum fed. So again, a very strong sex-dimorphic response to this. So our take-home for this is that we have long-term weight loss, which we associate as a positive benefit in the non-human primates using a relatively short-term intervention. So 2.5 months of the cyclical diet led to these really robust long-term positive effects in the males. We attribute it to changes partially in the metabolism. We're not exactly sure how returning back to the baseline is able to maintain this loss of body weight and improved what we believe health. And the females have now overcompensated. And this is essential, and we also believe it's tied to changes in the microbiome. So the take home for us as a group here and the translatability of this is, one, we don't need to start this diet necessarily at very young age. These interventions, as many people have mentioned before, we need to consider sex as a variable. They're not going to be consistent results. And the future directions that we'd like to do is, with the non-human primates, you definitely need to have a larger N. We're definitely interested in understanding the mechanisms that are driving these observations that I've just shared with you. And one of the things we're most interested in is these changes in epigenetic remodeling that may elicit this long-term memory. And also various other interventions that we can be using the non-human primate model to study how this may affect us as humans. So with that, I'd like to thank my team. Again, most of this work was done by Hagai Yanai and Bongsu Park. And very happy to take questions if I have time. Thank you.
Thank you. Thank you, Isabel.
Isabel, that was beautiful. I just noticed the caloric restriction in your group.
I told you, we're not very good at it, so we need other ways.
A question. So what the sex differences have been noted in humans who are intermittently fasting? Men are losing more weight and are maintaining more weight. So the question for me is, you showed that the women body weight went to normal, but did their food intake go to normal? And the reason I'm asking is the relationship between the effective CR and the body weight. In other words, did they have a set point where, because of the caloric restriction, they decrease energy expenditure, and that's why they could, they just maintain the same weight? That's
a fantastic question that we didn't address, and because we really were just maintaining the body weight over the time, right?
The food intake, you don't.
Well, during the time course of the experiment, yes, but the food intake over that 2-year time period was no longer as regulated. I mean, they got the same amount of biscuits, and it wasn't noted.
Experiments on those ones.
We did not.
Quick question.
Thank you.
Hi. Did you compare different time-restricted times, actually early time-restricted ones and late time-restricted ones, and which is better?
So we didn't do the time-restricted, so Joe might say, clearly this was a question. But again, in setting up this experiment, it really has to do with kind of how people-wise you can do this. So the way that the monkeys were fed first thing in the morning, like 6 a.m. So they all received their food at the same time, but we didn't do a restriction where we fed them, let's say, in the afternoon or maybe while they were sleeping or off cycle, because I believe you're asking questions of essentially during the circadian rhythm, when would be the best time to look at this. So one thing that we liked to maybe suggest in this is that it's over multiple circadian cycles that we're looking at here. So we may have avoided that, but it's a question that we didn't look at that is a very valid question. So sorry we didn't look at it.
All right, thank you so much, Isabel. Really fantastic talk. Thank you.